Probably all of us already met this situation in their daily lab life. An antibody staining is not working when buying a new lot, or staining with an antibody gives uncertain or even unlikely results. Fully characterized antibodies have been identified to be the main cause of the so-called antibody crisis. A Nature survey in 2016 revealed that 70% of researchers were not able to reproduce other scientists' experiments. However, reliable antibodies are essential for groundbreaking research. That's why this webinar or this talk will be about how to make excellent antibodies, or how we at Synaptic Systems make highly validated antibodies for histopathology. First, I will introduce who we are and the company's portfolio, which is divided into the SciSci and the HistoSure brands. Then I will show our antibody development and validation process to you, and this in more detail using our anti-F480 and anti-nucleocapsid antibodies as an example. I will introduce our species-specific antibodies, which we develop for application in chimeric tissues as used in preclinical oncology. And finally, I will briefly address how we use Multiplex IHC-P for antibody validation and how we enable multiplexing to our customers using our recombinant antibody technology. Synaptic Systems is a German company located in Göttingen in Lower Saxony. Synaptic Systems was founded in 1997 by well-known scientists of the Max Planck Institute in Göttingen due to a high demand for antibodies against synaptic proteins. This, of course, explains our name, Synaptic Systems. For 24 years, we are constantly growing and are around 25 people today, with a second dependence in Braunschweig, also in Lower Saxony, Germany. In 2017, we completed our original SciSci brand, which consists of antibodies against synaptic and neuroscience-related proteins, with our HistoSure brand, antibodies specifically developed for application in formalin-fixed paraffin-embedded tissues. Our mission is to provide excellent antibodies for cutting-edge science. To achieve this, we produce all our antibodies in-house and develop them in collaboration with leading scientists in the field. To better satisfy our customers' needs, we decided last year to present our two brands on two separate websites. On the Synaptic Systems website, you find our SciSci brand, which consists of approximately 2,500 antibodies and antibody-related products for neuroscience and cell biology. We offer polyclonal antibodies from rabbit, chicken, and guinea pig, and monoclonal antibodies from rat and mouse. Recently, we established a recombinant antibody platform to provide monoclonal recombinant antibodies with a rabbit, guinea pig, or chicken backbone. On our SciSci website, we offer also camelid single domain antibodies from our partner company, Nanotech. Our HistoSure website is tailored to the needs of customers working in histopathology. It includes, for example, a section on IHC-P guidelines and small articles on tissue-related applications. Our HistoSure brand consists of antibodies for use in formalin-fixed paraffin-embedded tissues for mouse pathology, species-specific antibodies for xenograft pathology, and antibodies for human pathology. Although the two brands are presented on separate websites, all products can order through one single web shop, and websites are linked to jump back and forth. Validation is the key issue when developing reliable antibodies. HistoSure antibodies are probably unique in that they are developed from scratch in formalin-fixed paraffin-embedded tissues. Especially in monoclonal antibody development, we are screening candidates from early beginning using immunohistochemistry. Specificity of our antibodies is tested in formalin-fixed paraffin-embedded cell pellets of cell lines with endogenous expression of the protein of interest or in transfected overexpressing cells. Of course, we use complementary validation methods to ensure specificity of antibodies, for example, Western blot, ICC, or IHC in vibrotome sections. Reproducibility, sensitivity, and potential non-specific binding of the antibodies is tested in antigen positive and negative tissues, and if possible, in high and low expressing tissues. For this, Synaptic Systems has built up a comprehensive tissue bank comprising around 700 blocks of murine, rat, and human tissues and paraffin-embedded cell pellets. Our HistoSure antibodies are validated according our HistoSure quality guidelines. This means that HistoSure antibodies intended for mouse pathology are tested in broad murine tissue panels and whenever possible by external collaborators. Species-specific antibodies are tested in appropriate human and mouse tissues to test for potential cross-reactivity. HistoSure antibodies that are intended for human pathology have the highest validation standards. Here, external validation by pathologists in clinical samples is mandatory. To do this, we have collaborators. Our main collaborators are Professor Dr. Feuerhake from the Medical School in Hanover, Dr. Pils from the Helmholtz Center in Braunschweig, and Dr. Schüler from Charles River Laboratories in Freiburg. To ensure maximal product transparency, we offer for each HistoSure antibody individual reference protocols that we used internally for antibody validation. These reference protocols are accessible through the product pages, where you can also find some background information on the target protein. And we give also individual protocols for ICC, Western blot, and IHC if the antibody is recommended also for these applications. In our IHC-P reference protocols, we use standard reagents from Agilent, Jackson Immunoresearch, and Vector Labs to ensure easy reproducibility. We use standard methods for antigen retrieval. This is heat-controlled, heat-induced antigen retrieval using a conventional kitchen food steamer. And we use short primary antibody incubation times of 1 hour at room temperature to enable easy adaptation to automated staining platforms. To show our validation process in more detail, I picked up F480 as an example, which is a macrophage marker for murine macrophages. Antibodies that we develop for mouse pathology are designed to mouse proteins for application in mouse tissues. This may be in contrast to other companies which usually develop antibodies that work in a broad range of species. This means that these antibodies are working also in mouse tissues, but may not as highly validated for murine application. We currently focus on macrophage markers and other immune cell markers that we develop in collaboration with Charles River Laboratories in Freiburg due to their demand for high quality antibodies for staining of syngeneic mouse tumor models. Our rat monoclonal antibody against F480 was highly validated in different IHC-P settings. For example, specificity of binding was tested in vitro differentiated murine macrophages, where an overall staining can be seen, whereas staining events in bone marrow of mouse is rare, reflecting the biological situation of low prevalence of F480 positive cells in this mixed population. Sensitivity Reproducibility of staining and potential non-specific staining was tested in a broad murine tissue panel. Here, mouse colon staining, liver, kidney, and lung staining is only shown as an example. Co-localization of F480 with IBA1, which is an alternative marker for murine macrophages, was tested in IHC in vibrotome sections and in IHC-P in murine mouse spleen. Final validation was done by Charles River Laboratories in syngeneic mouse tumor models using an automated staining platform. Our F480 antibody does not recommend proteinase K treatment and is therefore highly suited for multiplexing. Our antibodies that we develop for human pathology have even higher validation standards. To support the fight against the COVID-19 pandemic, we developed antibodies against the nucleocapsid protein of SARS-CoV-2. SARS-CoV-2 is a single-stranded RNA virus, and its nucleocapsid protein has only limited homology to nucleocapsid proteins of other coronavirus family members. Antigen specificity of our antibodies was tested in formalin-fixed paraffin-embedded cell pellets of cells transfected with full-length nucleocapsid proteins of diverse coronavirus strains, as shown here exemplarily for our antinucleocapsid antibody, which is specific for SARS-CoV-2. Mandatory final validation was done by Professor Krasemann from UKE Hamburg in lung tissue of deceased COVID-19 patients. The full validation data is accessible at our HistoSure website. For HistoSure, we put a special focus on species-specific antibodies as used in preclinical oncology. Here, conventional xenograft mouse models where human cell lines are engrafted into immunodeficient mice have substantial limitations, for example, in drug screening or in estimation of drug efficacy. To better mimic the human situation, PDX models are used where tumor samples obtained from patients are transplanted into immunodeficient mice. But still, this does not reflect the in vivo situation of the tumor microenvironment. So models can be improved by transplantation of human CD34 positive hematopoietic stem cells into immunodeficient mice, thereby establishing a human immune system. By additional transplantation of human fibroblasts, one can also mimic the non-immunogenic tumor microenvironment. However, biomarker research in these mixed models is hampered by the lack of antibodies which can distinguish between mouse and human cells or mouse and human proteins in these mixed backgrounds. To close this gap, we establish species-specific antibodies that can discriminate between human and murine cells. Validation of the species-specific antibodies are done in appropriate mouse and human tissues to test for potential cross-reactivity, and final validation is done in chimeric tissues, for example, classical xenografts or humanized mice that we obtain from Charles River Laboratories in Freiburg. We are currently developing antibody pairs for identification of human and murine immune cells in humanized mice. In this chromogenic double staining, our anti-human lamine B1 and anti-mouse lamine B1 antibodies can be used to detect human and mouse cells in this mixed xenograft model. For detection of proliferating cells from mouse and human origin, one can use our mouse and human-specific Ki67 antibodies as shown here in a humanized mouse spleen. We use Multiplex IHC-P mainly for antibody validation. Here, chromogenic and fluorescent multiplex IHC-P can be used to test for colocalization or non-colocalization of markers. F480, for example, colocalizes with CD163 in a subset of Kupffer cells in mouse liver, whereas the T cell marker CD3E does not colocalizes with the B cell marker CD19 in mucose-associated lymphoid tissue in a mouse colon. In our lab, we use secondary systems from Vector Laboratories for antigen detection in chromogenic IHC-P, either a combination of a horseradish peroxidase coupled impress secondary with the alkaline phosphatase coupled ABC reagents, or with a second impress secondary antibody coupled with alkaline phosphatase. For multiplexing, it is recommended to use primary antibodies from different species, but we will hear more about this in the following talk from Craig. However, multiplex IHC-P is not only relevant for antibody validation, it's also a very useful tool to answer scientific questions. To enable multiplexing to our customers, we established a recombinant antibody technology which enables a species switch of our monoclonal antibodies primarily produced in mouse or rat. To do this, we isolate the genetic information of our mouse or rat monoclonal antibodies conventionally produced in hybridoma cells and replace the genetic information for the constant region. Then the plasmids are transfected into maxi cells and we isolate stable antibody secreting cells. By using this technology, we can species switch an original rat or mouse monoclonal antibody to a recombinant antibody having a rabbit, chicken, or guinea pig backbone. Ideally, the species switched antibodies show identical staining patterns as the original monoclonal clone. As an example, here's IBA1, which is a macrophage marker in mouse and human, and a biomarker for activated microglia. As shown in this panel, our species switched rabbit and chicken recombinant monoclonal antibodies show identical staining patterns as the original mouse clone in mouse brain, in mouse spleen, and in human tonsil. So by using this technology, we can give or we can offer a broader diversity of primary antibodies to our customers, thereby facilitating their multiplexing approaches. If you want to learn more about our company, about our brands, and about our validation process, please visit us on the internet at either www.sci-com or at sci-histosure.com. Thank you for your attention.